Hello all, welcome to my channel Weight Lecture. Today we will discuss multiple choice questions on animal nutrition. Question number first. Vitamin K deficiency seen by the injection of option A sulfonamide drugs, option B tetracycline drugs, option C penicillin, option D all. The right answer is option A, that is sulfonamide drugs. Since vitamin K is itself synthesized by intestinal bacteria, and while on administrations of these sulfur drugs mainly cause deficiency of this vitamin K, either by decreasing its absorption or by decreasing its bacterial synthesis. Let's see some other facts related to vitamin. Vitamin B and C are synthesized in rumen. Similarly, vitamin E deficiency causes mulberry heart disease in pig. Mulberry heart disease is not so common disease in pig. It is a sporadic disease and mainly characteristic like that sudden death without showing any preclinical sign. And exotic diathesis in chicken, mainly seen in those chicken where we fed as torula yeast as a source of protein, which is causes the deficiency of vitamin E. And fatal syncope disease in pig and calf, characterized by sudden fainting of this animal, lead to that decrease in BP and then heart rate. Similarly, vitamin A deficiency cause xerophthalmia in pig, characterized by that dryness of that conjunctiva and cornea of eye. Okay, and biotin, which is vitamin H, is a part of acetyl-CoA carboxylase enzyme. Acetyl-CoA carboxylase enzyme play a vital role in the synthesis of fatty acids, which is used for the conversion of acetyl-CoA to that malonyl-CoA form. Okay. Liver of polar bear has very high amount of vitamin A. Similarly, vitamin D help in the synthesis of specific messenger RNA. Vitamin B2, that is riboflavin, consists of isoalloxazine nucleus. Similarly, that storage site of vitamin E is that I2 cells of liver, and then conversion of vitamin A from carotene is performed in epithelial cells of intestine. Carotene is that form of vitamin A which is available from in plant. Okay, one international unit of vitamin A is equivalent to 0.6 microgram of beta carotene. Also, vitamin A promote mucopolysaccharide synthesis by activating calcium molecule. Ascorbic acid, which is vitamin C, was first isolated by Jen Gorgi. Similarly, sheep don't need any additional vitamin A if that minimum carotene content in that ration is 5 mg per kg body weight per kg feed. Similarly, vitamin D stimulates incorporation of phosphorus pentaoxide into phospholipid of intestinal mucosa. Question number 2. Urea toxicity results when rumen ammonia level exceeds mg per 100 ml. Okay, in mg per 100 ml. Option A 60 mg per 100 ml. Option B 70 mg per 100 ml. Option C 80 mg per 100 ml. Or option D 100 mg per 100 ml. The right answer is option C, that is 80 mg per 100 ml. Okay, since urea toxicity is seen when that ammonia level exceeds 80 mg per 100 ml of ruminal fluid. Okay, let's see some other facts related to urea. Nickel is that essential for that urease activity of ruminal microbes, urease enzymes, which is used for the conversion of urea to the ammonia form. Okay, and the recommended label of urea in the total dry matter diet in dairy cattle is 1%, and the accepted percentage of total protein in diet of dairy cattle beyond which urea isn't benefited is 13%. Beyond exceed that 30% of a protein that urea isn't benefited in that dairy cattle. The bacteria unable to use ammonia effectively if ammonia concentrations in rumen per ml of rumen fluid per 100 ml of rumen fluid exceeds 5 to 8 mg per 100 ml. Maximum degradability of urea is 100%, that total urea can be degraded. 80% of nitrogens excreted 80% of nitrogen excreted in the form of urea in urine. since we are uricotelic, in case of uricotelic animal, okay, in urea cycle, ammonifications of carbonic acids is done. Okay, maximum permittable limit of urea in feed is 27 gram per 100 kg body weight. Okay, for 100 kg body weight. Similarly, urea doesn't feed in following condition. In case of young cattle, we should not feed urea, and similarly lactating and non-ruminant cases, because they doesn't utilize urea properly and it may cause urea toxicity too. In case of young cattle, that rumen is not completely developed, so we should not feed urea in these animals. Questions number 3. A silage containing animal organic waste, that is poultry drooping, swine excreta, is called option A, haylage, option B, wastelage, option C, brown silage, option D, bad silage. The right answer is option B, that is wastelage. A silage that containing animal organic waste, that is poultry drooping, swine excreta, is called wastelage. Let's see some other facts which is related to the silage. That is, silage is categorized into three categories. That is very good silage, which has pH range from 3.5 to 4.2. Similarly, good silage, which pH range from 4.2 to 4.5, and bad silage, which is the highest pH more than 5. Okay, since the main reasons for that deteriorations of that silage quality is that continual presence of oxygen, and the bacteria which allow to that actually propagate in that silage, which is lactobacillus, while coliform and clostridium bacteria shouldn't allow to propagate in silage. And in those cases where that silage undergo clostridial fermentations, we see that the butyric acid and acetic acid content is high in that type of silage. Okay. Commonest type of silage that produce from grasses where lactic acid bacteria is dominant. That type of silage is also known as that commonest silage and good silage, and that lactate silage too. Due to pentamethylene diamine adenine betaine extra, that characteristic of smells occurs in that silage. Okay, and at one time, after opening the silage, at the one time, layer of silage removed is 3 inches. And for the silage making, and the silage take minimum keeping time is at least 4 weeks. And height of that filling label of that silo is actually a pit in ground for silage making, and is 5 feet. Okay, plant for making that hay and silage should be cut at that flowering stage. And in case of that, there are two type of silage, that legume silage and non-leguminous fodder silage. In non-leguminous fodder silage, that already there is a content of carbohydrate is high, and so there is no need of additional molasses added. But in case of that leguminous silage, we should add that additional molasses, so that it helps in fermentation process too. Okay, the temperature of that silage is 38 degrees Celsius, and that pH of the silage is 4. Question number 4. Desired calcium phosphorus ratio for dog is option A 1.2 to 1.4 is to 1, option B 1.2 is to 2, option C 1 is to 2, option D 2 is to 1. The right answer is option A, that is 1.2 to 1.4 is to 1. Desired calcium phosphorus ratio for dog is 1.2 to 1.4 is to 1. Similarly, the desired calcium phosphorus ratio for cat is 1 is to 1. And the desired calcium phosphorus ratio in cow milk is 1.2 is to 1. Question number 5. Taurine is essential amino acid in option A, cattle, option B, horse, option C, pig, option D, cat. The right answer is option D, that is cat. Taurine is essential amino acid. In case of that cat, we should supply the taurine from externally to that cat. Similarly, wheat is deficient in lysine amino acid, while pulses are deficient in methionine amino acids. And cysteine can be synthesized from methionine. And that is first limiting amino acids in protein of groundnut cake, that is lysine. Limiting amino acid is those amino acids which is as compared to the standard protein, which amount is very low in that protein. Such type of amino acid is known as limiting amino acids. and we should supply this limiting amino acid from externally. Okay, in limiting amino acid in case of meat, bone, that is tryptophan. Limiting amino acid in case of soybean is methionine. Limiting amino acids in wheat, that is lysine. Okay, the percentage of non-essential amino acids in our body, that is 40%. 40% of amino acids not need to supplied from feed, body itself prepared in body. Okay, bile acids have cysteine and glycine amino acid. Lysines and arginine are antagonists of each other, that the absorption of one amino acids uh, that hindrance by that another amino acids. Similarly, increasing order of toxicity of amino acid, that is threonine, and that tyrosine is more toxic than threonine, and that methionine is more toxic than threonine. Methionine is that highest toxic amino acid. Okay. Example of ketogenic amino acid, that is those amino acid which gives ketones bodies like phenylalanine, isoleucine, tyrosine. While true ketogenic amino acid, that is leucine. Total type of amino acids which is present in body is 20, out of which number of essential amino acid which is need to be supplied from feed, that is environment, is 0, because almost all amino acids are prepared in that ruminal body itself, since the rumen get this amino acids from that bacterial protein, while in case of pig and human, that is 8. Okay, optically inactive amino acids, example is glycine and glutathione. While glutathione, while in tryptophan is optically active, since optically inactive is due to that absence of that chiral carbon atom, and while optically active nature of that amino acid is due to the presence of that chiral carbon atom. That chiral carbon atom is those carbon atom which consists that which four bond is attached with different four atoms. Okay, bond which is present between two amino acid, that is known as peptide bond. And since amino acid show that both acidic as well as base nature, so in alkaline solution amino acid act as an anion, while in acidic solution it act as a cation. A solution in which that amino acids pH is electrically neutral, that type of solution is known as that type of pH is known as that isoelectric pH. And amino acids are that soluble in water and insoluble in organic solvent. Question number 6. The number of KOH, potassium hydroxide, required to neutralize the free organic acid in 1 gram of fat is called as option A, acid value or number, option B, saponification number, option C, iodine number, option D, rancid number. The right answer is option A, that is acid value or number. The presence of free fatty acids in fat, which actually cause that rancidity of fat, called acid value or number. Since that rancid number or that peroxide value number is that the peroxide value, that is milli-equivalent peroxide, MEQ, per 1 kg of fat, 1 kg of fat, this is the peroxide value. When it is more than 7, we see that rancidity. Similarly, saponification number denote the weight of that KOH in milligram needed to saponify 1 gram of fat. And while iodine number is the number of iodine in milligram required to saturate the fatty acids which is present in 100 mg of oil or fat. And since the high grade carcass have that iodine value is that less than 65. Question number 7. Circulatory label of glucose in pig is option A 40 to 70 mg per 100 ml blood, option B 20 to 30 mg per 100 ml, option C 10 to 20 mg per 100 ml, option D 80 to 120 mg per 100 ml. The right answer is option D, that is 80 to 120 mg per 100 ml of blood. The circulatory level of glucose in case of pig, that is 80 to 120 mg per 100 ml of blood, while in case of cow is 40 to 70 mg per 100 ml of blood. Other facts which is related to glucose is that like glucose is also known as aldose sugar, dextrose, sugar of life, grape sugar, while fructose is known as fruit sugar or sweetest sugar. Question number 8. Which of the following is amino polysaccharide? Option A, starch. Option B, glycogen. Option C, heparin. Option D, fructose. The right answer is option C, that is heparin. Heparin, hyaluronic acid, chondroitin are example of amino polysaccharide. Similarly, glycogen, starch, fructan are example of non-amino polysaccharide. Glycogen is example of homopolysaccharides, and example of triose sugar, that is glyceraldehyde. Example of pentose sugar is arabinose. Example of hexose sugar, that is glucose, erythrose, galactose, etc. Question number 9. In dog, the amount of water lost through urine per day is option A 0.6 liter, option B 6 liter, option C 5 liter, option D 0.8 liter. The right answer is option A, that is 0.6 liter. The amount of water that lost through urine in case of dog is 0.6 liter. Similarly, the amount of water that lost through urine in horse is 5 liter, and the amount of water that lost through urine in cow is 7 liter. It is the data that the amount of that water lost through urine per 24 hour. Question number 10. Enzyme carbonic anhydrase has which element? Option A, iron. Option B, zinc. Option C, magnesium. Option D, copper. The right answer is option B, that is zinc. Enzyme carbonic anhydrase has zinc. Since carbonic anhydrase enzyme is used for that the reversible conversion of that carbon dioxide and hydrogens into that carbonic acids and bicarbonates, and that and vice versa. And this enzyme carbonic anhydrase has zinc, and this zinc is derived from that metabolic process of kidney. And zinc deficiency in case of like poultry causes Solon-Hock syndrome, while zinc deficiency in pig and cattle causes parakeratosis, that is incomplete or improper deposition of keratin on outer layer of the skin. Question number 11. Amount of iron excreted in sow milk. Option A 7 mg per day. Option B 8 mg per day. Option C 10 mg per day. Option D 12 mg per day. The right answer is option A, that is 7 mg per day. Amount of iron that excreted in sow milk is 7 mg per day. Similarly, that requirement of iron in case of pig is 15 mg per day, while in case of chick is 9 mg per day. And excretion of iron occur in case of hematuria, hemoglobinuria, menstruation, etc. Iron deficiency causes microcytic normochromic anemia in case of calf, while microcytic hypochromic anemia in case of pig and chick. 0% iron is actually excreted in case of urine. In feed, that iron is present in form of ferric form, while for that iron absorbed through GI tract as form of ferrous form, and the pH of that low pH of that GI tract help in the conversion of that ferric form into that ferrous form. Okay, iron absorbed through that intestine by active transport, that is, it need energy for its transportation. And iron present in storage tissue as in the form of hemosiderin. Similarly, iron present in the form of ferritin in that mucosa of intestine. And in muscles, iron presents as myoglobin, while in plasma, iron presents as a transferrin. And in body, most of that iron presents in the form of hemoglobin. Question number 12. Empirical formula vitamin B12 is option A, say 63H88O14 PCO, B C63H84O14 PCO, C C60H84O12 PCO, D C63H80O10 PCO. The right answer is option A, that is C63 H80 O10 PCO. Empirical formula vitamin B12 is C63 H80 O10 PCO. Empirical formula vitamin E is C29 H50 O2. Empirical formula of carbohydrate is CnH2O whole power n. Question number 13. Crude protein in crib ration of swine is option A 10%, option B 13%, option C 16%, option D 18%. The right answer is option D, that is 18%. Crude protein person's requirement in crib ration of swine is 18%. Crib rations means like those feed which is supplied to those baby suckling pig. Okay. Required percentage of crude protein in case of grow ration of swine is 16%. Required percentage of crude protein in boar ration is 14%. And said since that out of whole total cost 75% of total cost is related to that feed cost in case of swine. Volatile fatty acids, since swine is non-ruminant, in case of that non-ruminant, that fermentation process mainly, if fermentation process seen, is mainly seen in case of siccum. So volatile fatty acids are produced in siccum of swine. Nutritive ratio of starter is 1:4. It's a ratio of that the protein upon that other nutrient in that feed. First limiting amino acid of a pig is lysine. 1 kg total digestive nutrient in swine feeding equal to 4500 kcal of energy. Diet of pig should be supplied with linoleic acid. Pellagra is a disease which is mainly characterized by 3D, that is dementia, dermatitis, and diarrhea. In pig is mainly caused due to deficiency of niacin, that is vitamin B3. Goose stepping walking caused due to deficiency of both vitamin B3 and B6, pyridosins and niacins. Folic acid deficiency in pig cause both anemia and that deficiency of lipid, that is lipopenia too. Diet of pig should be based on the basis of concentrate. Similarly, energy requirement of a pig is expressed in terms of digestible energy or metabolic energy, kcal per kg of ration. Mineral requirement of swine depend on protein. Hog should be self-fed, except in case of the gestation time and breeding board. Starch cannot be fed to swine up to 2 weeks of age. Okay. Level of iron sulfate in case of creep feed is 2%. Level of crude fiber in diet of gestation sow is 10 to 12%. And average amount of feed that consumed by adult pig is 3.5 to 4.5 kg per pig per day. Feeding schedule for the potential porker and heavy hog up to 45 kg body weight is that adlib allowed to feed at any time when they want. Question number 14. Level of Rovi mix in poultry diet is option A 150 gram per quintal of feed, option B 200 gram per quintal of feed, option C 250 gram per quintal of feed, or option D 300 gram per quintal of feed. The right answer is option C, that is 250 gram per quintal of feed. Label of Rovi mix in poultry diet, that is 250 gram per quintal of feed. Rovi mix is like superior formulation of vitamin E, and similarly label of Rovive in poultry diet is 20 gram per quintal of feed. Rovive is none other, that is a formulation which contain vitamin B complex. Pearson square concept is used in poultry nutrition to calculate protein. Similarly, yeast should be fed in case of zinc deficiency. Chelating agents such as EDTA are sometimes used in chick ration in order for increasing zinc absorption. Required level of iodinated casein in poultry diet is 110 to 220 mg per kg of feed. Poultry nectar is used as flavoring agent in poultry nutrition. Sodium bentonite is used as pellet binder in case of poultry nutrition. Safety margin for vitamin in case of poultry ration is twice. We can we can increase vitamin supplement to that poultry up to its twice concentration. It's safe. Poultry rations should have 1% linoleic acid and 0.01% antioxidant and 2.3% calcium. Basal's metabolism productions of poultry is 2.75 calorie per kg live body weight per hour. For 0 to 6 week old chick, methionine should not be less than 0.46%. Okay. Similarly, requirement of vitamin A in poultry is 800 international unit per kg of feed. Only essential fatty acids of poultry, that is linoleic acid, which we should need to supply from outside. Manganese required in maximum amount in poultry ration. Calcium and phosphorus ratio in boiler finisher is 155:1. Protein percentage in boiler ration is 90 to 24%. Uh, Poultry should feed its in limit to satisfy its energy needs. Feeding requirement of a poultry is expressed as kg of feed per 100 birds for 100 birds. Optimum level of fat in poultry diet is 2 to 5%. Percentage of total expense in poultry feed, that is 60 to 65%. Question number 15. Crippling disease occur due to option A, genistein, option B, chaumerism, option C, lethyrogen, option D, mimosine. The right answer is option C, that is lethyrogen. Crippling disease or lethyrism, that is a neurotoxic disorder which mainly affect like people and domestic animal by characterized by irreversible spastic paralysis of hind limb, so animal unable to walk properly. While genistein is a phytoestrogen that occur naturally in the diet, especially in soybean and soy-based food, and it's mainly an interest of for that chemo preventive agents for various variety of disease and cancer. Mimosine toxin found in that Mimosa pudica is mainly characterized by alopecia, anorexia, weight loss, hypersalivation, low thyroid concentration, etc. Question number 16. Digestibility of treated straw is option A 46%, option B 70%, option C 80%, option D 30%. The right answer is option B, that is 70%. Digestibility of treated straw is 70%. Digestibility of untreated straw is 46%. Since straw can be treated by with like alkali, acids and salt all. 4% calcium hydroxide is used for about 4 months for straw treatment. Similarly, 12% potassium hydroxide is used for 4 months in straw treatment. 2.5% of ammonium hydroxide is used in straw treatment. And note, since on grinding of that grain, the digestibility increases, while in case of that rough edges hay, the digestibility on grinding, the digestibility decreases. Question number 17. The normal bacterial population per ml of rumen content is option A 10 power 9 to 10 power 11, option B 10 power 6, option C 10 power 17 to 10 power 18, option D 10 power 15. The right answer is option A, that is 10 power 9 to 10 power 11. The normal bacterial population per ml of rumen content is 10 power 9 to 10 power 11 per ml of ruminal content. The normal protozoal population per ml of rumen content is 10 power 6 per ml. Protozoa cannot tolerate a pH which is less than 5.5, and microbial protein have that biological value of 0.8. Under a normal condition, pH of ruminal liquor is 5.5 to 6.5. The normal ruminal flora is established at age of 6 weeks. In total, digested microbial protein, the protein percentage contributed by protozoal origin is that 25%. Since immediately after a meal, the gas productions in that rumen may exceed 30 liter per hour. Mineral that cannot be absorbed by rumen, that is calcium and magnesium, while sodium, potassium, cobalt, iron are absorbed from ruminal wall. Note that sodium and potassium absorbed against concentration gradient. Rate of lactate absorption in rumen is 1/10 of that rate of that volatile acids absorption. Volatile acids absorbed in rumen in the form of anionic form. Total volatile fatty acids in rumen is 60 to 120 milliequivalent per liter. Percentage of methane gas in rumen is 30 to 60%. Question number 18. Loss of dry matter due to effluent is option A 20%, option B 2 to 10%, option C 10 to 60%, option D 60%. The right answer is option C, that is 10 to 60%. Dry matter loss due to effluents is 10 to 60% of dry matter is lost due to effluents. Effluents is that like a leaching form, like in sewage in case of that water loss, liquid waste, that is not effluents. And similar, the loss of dry matter due to anaerobic fermentation is 2 to 10%, and 20% of dry matter is lost due to aerobic fermentation. Question number 19. Debranching enzyme in glycogen synthesis is option A amylo 1,4 glucosidase, option B amylo 1,4 glucosidase, Option C, amylo 1,3 glucosides. Option D, amylo 1,2 glucosides. The right answer is option B, amylo 1,6 glucosides. Debranching enzymes in case of like glycogen synthesis is amylo 1,6 glucosides. Similarly, branching enzymes in glycogen synthesis is glycosyl 4,6 transferase. Branching is more common in case of glycogen than starch. Degradation of glycogen is known as glycogenolysis. Synthesis of glycogen is known as glycogenesis. Degradation of glucose is known as glycolysis. Synthesis of glucose is known as glucogenesis. Vitamin B12 is participate in gluconeogenesis. Gluconeogenesis from like lactate and propionate is more common in case of like ruminant. Your TCA cycle occurs in mainly in mitochondria. Similarly, key molecule for the TCA cycle is oxaloacetic acid. And the percentage of creatinine. Creatinine has ATP stored in muscles in the form of creatinine. In skeletal muscles is 0.5%. And product of that anaerobic glycolysis is lactate and ethanol, while that the aerobic glycolysis is pyruvate. When CAMP, cyclic amino monophosphate, increases, glucose synthesis also increases. Enzymes which is present in liver but absent in muscles, that is glucose 6 phosphatase. Questions number 20. For glucose, which organ is insulin dependent? Option A, lungs. Option B, liver. Option C, brain. Option D, spleen. The right answer is option C, that is brain. Brain is that organ which depend on insulin for glucose uptake. Glucose gate of cell is opened by like insulin. Okay, the blood glucose level in laying hen is 139 to 290 mg per 100 ml of blood. Similarly, blood glucose level in non-ruminant is 30 to 120 mg per 100 ml. Similarly, blood glucose level in ruminant is 30 to 70 mg per 100 ml. Question number 21. Which of the following is used after defluorination? Option A, slake lime. Option B, alum. Option C, rock phosphate. Option D, none. The right answer is option C, that is rock phosphate. Rock phosphates are used after defluorination. Defluorination is a process of that removal of that fluorine from that the water content, and then rock phosphates are used after that defluorinations. And for the defluorination process, some other facts about fluorine, that's minimum permissible limits of fluorine in water is 3 ppm part per million, and excess of fluorine cause fluorosis, while deficiency of fluorine causes dental caries. Question number 22. Vitamin H, that is biotin, is also known as anti, option A, anti-angial factor, option B, anti-infective factor, option C, anti-aguide injury factor, option D, anti-inflammatory factor. The right answer is option C, that is anti-aguide injury factor. Vitamin H, that is biotin, is anti-aguide injury factor. Since egg injury is mainly caused by the like avidin, and which is removed by that vitamin H. Vitamin B6 is known as anti-dermatic factor. Vitamin K known for anti-hemorrhagic factor. Vitamin A known as anti-infective factor, and vitamin D is known as anti-rickettsial factor. Question number 23. Condensation enzymes of lipogenesis is which of the following? Option A, acetyl-CoA. Option B, beta-ketoacetyl ACP synthesis. Option C, acyl-CoA. Option D, none. The right answer is option B, that is beta-ketoacetyl ACP synthesis. Condensation enzymes of lipogenesis is beta-ketoacetyl ACP synthesis. Since like lipid synthesis or like fatty acid synthesis include following steps. First is like activation of acetyl-CoA, and second is that elongation of fatty acids, and the third is like termination of fatty acid synthesis. Since like in case of that elongations of fatty acids, it's include major steps: condensations, reductions, dehydrations, and again reductions steps. So in case of that condensation enzymes of lipogenesis is beta ketoacetyl ACP synthesis. Okay, for elongations, malonyl CoA is used in case of endoplasmic reticulum, while acetyl CoA is used in case of mitochondria. and that's the major sites of lipid fatty acid synthesis lipogenesis is liver and adipose tissue in case of cattle, only adipose tissue in case of pig, and only liver in case of poultry. Adipose tissue has only 10 to 20% of total fat, while 50% of total fat report its presence in subcut tissue. Synthesis of fatty acids occurs in case of cytosol, and beta oxidation, that's a breakdown of that fatty acids. Beta oxidations except activations occurs in mitochondria. Activations enzymes of beta oxidation is thiokinase. In the diet, ratio of anti-ketogenic food to that ketogenic food should be less or equal than 2. Hormone which is responsible for decreasing blood fat content, that is thyroxine. In case of like beta oxidation is a process of breakdown of fatty acids. Lipogenesis is a process of that formation of fatty acids, okay? And the cleavage enzymes for beta oxidation is beta ketothiolase. Beta oxidations was first discovered by Noob. Beta hydroxybutyrate and acetoacetic acids are used as a source of that energy preferable by skeletal muscles. Example of ketone body, that is acetate, acetoacetic acetates, beta hydroxybutyric acids. Level of acetate in blood, that is 10 mg per 100 ml. Question number 24. Single article of food is known as option A feed, option B ration, option C diet, option D none. The right answer is option A, that is feed. Single article of food is known as feed. Amount of feed that offered to an individual for a specific period, that is known as ration. And that amount of feed consumed by animal in a given time, that is known as diet. Okay. Question number 25. Color of bile juice in cattle is option A green, option B dark yellow, option C golden yellow, option D blue. The right answer is option A, that is green. Color of bile in case of ruminant is green due to presence of biliverdin, while color of bile in case of non-ruminant is golden yellow due to presence of bilirubin. Question number 26. Concentrate that fed to cattle have how much TDN, total digestible nutrient? Option A 65 to 68%, option B 68 to 72%, option C 72 to 75%, or option D 76 to 79%. The right answer is option B 68 to 72%. Concentrate that fed to cattle should have total digestible nutrients 68 to 72%, and then the production rations have concentrate in crossbreed is 1 kg per 2 for 2 kg of milk production, while production ration in Jebu cattle is 1 kg for 3 kg of milk production. Concentrate that fed to cattle should have 14 to 16% digestible crude protein. Similarly, requirement of digestible crude protein at 5 months of pregnancy is 0.14 kg. Carotene, which is a source of vitamin A, should be fed to cattle 4 times more than required rate. Dry matter requirement for camel is 2% of their body weight. Similarly, dry matter requirement in buffalo feed is 2.5 to 3% of their body weight. Dry matter requirement Jebu cattle is 2 to 2.5% of their body weight. Amount of succulent in cattle feed, that is 2/3 part of the total roughages is that succulent, and 1/3 part is the dry roughages. Level of mineral mixture and salt in cattle feeding is 1% of total concentrate. Okay, for 1 liter of milk production, requirement of mineral is 0.7%. Question number 27. 1 degree Celsius increases vessel's metabolic rate up to option A 20%, option B 30%, option C 33%, option D 40%. The right answer is option C 33%. 1 degree Celsius increases vessel's metabolic rate up to 33%. Since vessel's metabolic rate is that number of caloric burn just to perform life sustaining activity. Okay. Questions number 28. Pepsin isn't an example of option A exopeptidase, option B endopeptidase, option C both, option D none. The right answer is option A, that is exopeptidase. Okay. Since peptidase is an enzyme which is used for that breakdown of that peptide bone first, and exopeptidase break that terminal peptide bone, while endopeptidase break middle peptide bone, internal peptide bone. An example of that exopeptidase is that carboxypeptidase, aminopeptidase and dipeptidase, while example of like endopeptidase is pepsin, trypsin, parapepsin. And since aminopeptidase and dipeptidase are peptidases of intestine and also known as erepsin. Question number 29. Which of the following hormone synthesis is banned? Option A, stilbestrol. Option B, insulin. Option C, testosterone. Option D, thyroid. The right answer is option A, that is stilbestrol. Since estradiol or its analog has a carcinogenic effect, now it is banned to use as growth promoter. And hormone which is used to increase milk productions are thyroxine and growth hormone, and hormone are not used as a stimulator in case of like breeding and pregnant animal. Questions number 30. Hay that stored at high moisture content more than 20%. Option A, show marked temperature increase due to growth of thermophilic mold. Option B, increase the digestibility of nutrient. Option C, increase the wastage of hay. Option D, all. The right answer is option A, that is show marked temperature increase due to growth of thermophilic mold. Hay that is stored at high moisture content more than 20%, so marked temperature increases due to growth of thermophilic mold. In the preparation of good quality hay, following things should be considered. Option A, first, green fodder shouldn't expose to sun for longer period without proper turning. Second, the fodder should not be exposed to rain. Third, rapid sun drying that help in formation of vitamin D2 and conversion of carotene. Okay. Okay, class, this much for today, and I will come with another topic in another video. Okay, thank you.